Good. Well, hello, everybody. It's me, Dennis Hanasak. I hope you all can hear. Not, send us a quick note, but I think everybody should be able to hear me okay. There's still some people coming in, but we got the vast majority of the people are here. Couple of things. If you're wondering why I am masked, it's because I'm here at Harvard Ranch, and there's 4 of us in the room, besides myself, David Uberger and Kristen Davis, who work on the project, and I'll talk about them a little bit later. And Wyatt Kudo, who you will see is our host. He is our IT excellent person who's going to make sure everything goes smoothly, and he's helped us with the surveys and so on and so forth. I hadn't planned to talk about the agenda, but since there's a couple people coming in, I'll just do a real quick overview. I'm going to give you an overview of what our overall effort is in regards to developing a monitoring plan. Dwayne DeFries will come on and discuss how this ties in to the NEP and its CCMP. And then we're going to have 3 talks, all by members of our steering committee, which is really the crux of the information that we're trying to get to you or share with you today. And that's going to be Chuck Jacoby, Jeff Beal, and Derek Cox. And they're talking about different parts of the vital signs that are called out in the CCMP. We'll take a break then. At that point, I'm thinking we'll all need a good break. And then I will go over a draft list of recommendations that came about from the steering committee. And we throw them out to you to see what your reactions are. And then at the wrap up, I will primarily share with you some of the results of the surveys that we'll be doing during those 3 presentations that I mentioned earlier. So we're going to try to get some very quick feedback from you all that we think is going to be really critical. I encourage people to actually turn on their videos. I know a lot of times people don't want to do that. They say, bandwidth problems, yada, yada. Well, we also know there's other reasons for doing that. But we think that if you really, we would have done this in person. That was the preferred way. And what we tried to do is to make the meeting shorter, more concise, more interactive, like with the surveys. And I think to the extent possible, I don't know, we've all been on a lot of these things. I just find it's a lot friendlier, and I find that I stay a lot more focused when I can see my colleagues out there. So feel free to turn on your video. Also, the chat room is open, and you should, throughout the course of the meeting, if you want to ask questions, that's the best way to do it. David will be monitoring the chat the whole time. And also, if you just want to say things that you want us to ponder or you want everybody else to see. Basically, as you know from my email, we are recording this. It's going to be made available to a lot of other folks to look at. But the chat for us is going to be a great resource to get, your real-time feedback. And to the extent possible, when it is a question directed to one of the speakers, we'll try to answer that during the time we have, or we will encourage the speaker to continue this discussion in the chat, and everybody can see what that is. Nothing is going to be lost from all of this input, and that's an important thing. Okay, so with that, I think we've allowed enough time for some of the stragglers to get in. It's 1:05, and if you can set me up for my presentation. So I think you all know that this work is funded through the NEP. So thank you, Dwayne, and all the staff. And it's one of several projects that the NEP is funding that I would say falls under the category of planning for the future. And you'll probably hear a little bit more about those other projects as well during the course of the day. I wouldn't be surprised if that didn't come up. And really, the purpose of our workshop today is to really talk about that monitoring plan, an important step as we start to write what this final report will look like and what our recommendations will be. That report will be vetted by a lot of our other people. It'll be posted just like the CCNP was on the IRLnet site for comments. And Dwayne will help us get through that procedure pretty well. Now, the purpose of this, in general, as I said in the advanced information you got, is really to take a look at what's going on currently and develop a comprehensive lagoon line monitoring plan. What do we have asset-wise? Can we identify gaps of any type? What are emerging needs? Things that we could be doing better? Specific recommendations for action. Who are we targeting this to? Well, first and foremost, and most of the people today are in this category, which is the data collectors and managers. The idea is hopefully to get feedback to improve the overall monitoring system. But also an important part of this will be sharing it with the public, because in the end, we have great accountability on what we do. Most of us work one way or the other with public dollars. And of course, the lagoon is a very high priority for everybody who lives along it and probably beyond. I want to introduce our project team. I already mentioned them to let you know why we were being, why we're here in rural Mass. This is our guidelines. I think a lot of you know me, and I work, I'm really basically started out as a plant guy. I worked with macroalgae and seagrasses and water quality. Got into doing some crazy stuff with online monitoring. And so I've always had this interest in monitoring, really from a scientist point of view, though, trying to better understand, how the organisms and how they respond to it. David has been working with me for a couple of years, and he's doing a lot of great things. He's the one that worked with all of you who responded to our questionnaire, and he's put together all the tables and the maps that you're going to see. And one of the things that really attracted me to this project was that I knew David previously had a lot to do with the revision of the Aquatic Preserves Management Plan. And that was the first time that was done, I think, in about 3 decades. And I remember reading that and attending some meetings about it. And I just was super impressed. So when David started working here, I said, we should figure out how to take advantage of that great skill he has. Kristen's worked with me a little bit longer, about 15 years, with water quality monitoring and seagrass work. And she's developed a lot of other expertises that have to do with data and data management and GIS. So you can kind of see how that also is a nice skill set to have here as well. Very importantly was, yes, this is a project funded to me as the principal investigator here at Harbor Branch, but in the proposal and in reality, we reached out to a large number of people up and down the lagoon who do monitoring or who are in management, people who use the data, need the data. And you can just look at that list, and I will just thank them probably hopefully more than once today, but they have really been truly helpful. I've tried real hard not to really steer the monitoring plan as much as try to get people on the steering committee who could really help us decide where things should go. And now we're expanding that group to include all the people involved today, because now we think we're ready. We have kind of a trial balloon, I like to call it always. Let's see how this looks, see what everybody else thinks. But really, the folks here on this list deserve quite a lot of credit. And you're only going to hear from 3 of them, but in most cases, their presentations were influenced strongly by other folks in the last couple of weeks. So the outcome is a comprehensive, coordinated, integrated monitoring network. I hope you're all familiar with the CCNP. If not, I'm sure that Dwayne can answer any and all questions, and we'll talk a little bit about this. But there's different groups of vital signs, and the ones that we were asked to focus on fall under the categories of water quality, habitat quality, and living resources. And so you'll see that a lot of our thinking and a lot of the way we structure, like the tables you saw, the way we're going to structure the report, the way you're going to hear the presentations today, we are totally following the approach of how does this directly link to the CCNP, and in particularly these vital signs. Each vital sign has a number of lanes, if you will, underneath them. Another thing is that all of this existing monitoring, David has been getting a tremendous amount of information from everybody who's replied to our questionnaires. And everything will be on a GIS map, and they will be readily available to everybody. Everything will be provided through EarlNet. Identifying gaps in the network, very important. Identifying new opportunities in emerging technologies, things that we haven't really used yet, or things that are just starting to be used that's really going to revolutionize what we do, say, in the next 10 years. I honestly don't believe I need this slide. Maybe I'll skip over it real quick. But why do we need to monitor? Well, I think maybe we just need to remember that in order to really be successful, whether you're a management or like myself, a scientist, we really need lots of different types of ecosystem observations. And more, with the opportunities we have now with big data, although most of us are not really using big data yet, but we will. But with all the opportunities that we have, environmental observatories is more and more the way to go. We need to understand that there's differences between, say, a sampling program. So I, as a scientist, might come up with a really cool project. And yes, it might involve measuring a number of parameters in a lagoon, but it might be, say, to address a very specific question, or maybe just happens to be a graduate student's thesis, and it's going to last just for that duration of that one project. It's very much a research-focused thing. So we want to really look at looking at more effective, more efficient ways to do things, provide the best data for decision making, and interpret data appropriately, and get those interpretations and caveats out so people understand them. So there's a difference between sampling efforts per se and long-term, what I'll call long-term monitoring efforts. And so again, the emphasis is on key indicators, times and places, and talking about how are we going to store the data and use the data is also, I think, under the auspices of discussing, what the monitoring program should be. I think we all recognize what adaptive management is, and I think we understand that so many things have happened. If we just look back 10 years ago, well, how many people thought there would be a brown tide 10 years ago? We didn't have a brown tide 10 years ago. When we first had the super bloom, it was called the super bloom because it was so unusual. That super bloom has been eclipsed by lots of other blooms since. So there's a lot of things we don't understand. We do understand now, probably more so than ever, that it's a very complex system and that its responses to stressors and management is something that's going to require a real monitoring program that's comprehensive and integrated. So what do we do with it? Well, this should drive adaptive management by evaluating what is happening versus what we thought was going to happen when we started to try to manage for something. We need to be able to adjust our management actions and probably try new things, new types of sampling. And I already said before, there are new ways that most of us are not yet using, but we will, and we should be. And also, it's really important that we be open and transparent with everybody, not just ourselves, but with the public. So, key elements. These are like this is like the cornerstone of how we came up with what we came up with. I think I've already said plenty enough about this one. It needs to be linked to management, okay? It specifically needs to support the CCNP and the vital signs. It should be something that is actively going on, as opposed to something that used to go on or something that might go on, okay? It should be active, it should be ongoing into the future. So this is about the future. It's not about everything that happened in the past. There's other projects that the NEP is supporting that will look at a lot of that legacy data. It should have a QA/QC program of some type. Oh God, just got some message about wait an hour or restart now. I think, no, let's not restart. So, okay. Data accessibility. We believe strongly that the data need to be freely accessed to all those who need to use it, who want to use it. We do realize that it's a tricky thing sometimes in terms of scientists and the need to publish, and I certainly understand that as well as anybody. I want to remind everybody that this plan is, the geographic scope is the IRL boundary, which includes nearly all of the IRL watershed, and in the last several years also includes the IRL Halifax River planning boundary. So we reviewed and added to an initial draft of major sampling and monitoring programs that we came up with a bunch of tables. You saw one table that summarized that from a questionnaire. And this is the types of stuff that we got. And we have nice little inputs from a lot of different people, a lot of different programs. And all that will be captured fully in the report. So everything that came into us will be in the report as an appendix, and we will tap into it as need be. The key questions that we're asking is really who, what, where, when, and why. So who is collecting the data? What type of data is collected? Some of the details are listed there. Where are we collecting it from? Location, the frequency of collection, and again, very importantly, how is this data linked to a vital sign or some management action? So today, what are we doing? I mentioned while I was stalling for more people to come on about the series of presentations that we're going to have about what we've learned so far. We're going to use the real-time polling in 3 of the sessions. I will try to summarize that really quickly at the workshop wrap-up at the very end. So stay till then if you want to know what y'all thought. And a reminder that the workshop is being recorded and will be available on the website. And we will be posting a final report. And I'm sure that prior to that, we will send it all to you to give your comments on, because we really value that you took the time out today, and therefore we think you have a vested interest in seeing that report before it goes to everybody else. So I think you will be part of that process. So I did that in record time, 13 minutes. So if there's any quick discussions or questions, I think David will let me know. We're good. Okay, then without further ado, Dwayne DeFries is going to talk to us about, he's going to give us a charge, linking the monitoring plan to IRL NEP and its CCMP. Dwayne.
Thanks, Dennis. And before I begin, I just want to say thank you. Dennis, his team at Harbor Branch, and the steering committee have been working really hard through what has been a really COVID challenging year for all of us. So kudos, Dennis, to you and your team and everybody who's been meeting along the way to get us to this point. And we've got 50 people on this call. And so we're going to be looking to each of you to weigh in and be actively engaged. And I just want to thank you all. I can't stress enough the importance of a monitoring plan in the context of a CCMP. So I'm going to just quickly put it into a larger perspective that some of you are aware of, but maybe not all. So we are authorized as a national estuary program in Section 320 of the Clean Water Act. And there's like 7 authorization directives to our NEPs. And more than a few are directed towards, tracking, whether the CCMP is working, how water quality is changing. So looking at status and trends, using monitoring as a basis. And each of the 28 designated NEPs around the nation all do this a little bit differently. There's a couple that are spending millions of dollars a year where monitoring is actually housed within an NEP. And I'm going to be honest, our NEP for its first 25 years, pretty much, was not engaged directly in monitoring per se, but we were engaged in supporting many of you who monitor And over the last several years, we have really increased our effort in that regard, where we are now doing recurring funding of the atmospheric deposition station. We're doing recurring funding of the harmful algal bloom group. And so we are engaged in monitoring. And so what you do in this plan is important because it's going to guide how we move in the future and where we move. But it's also important from a national standpoint with EPA. Part of our review process, so we can continue to get federal funds, is to meet obligations under 320. And so every 5 years we have a program evaluation. And there's 5 planning documents that the EPA calls consensus documents, which means that there is concurrence and consensus to the CCMP. The monitoring plan is one, a communication plan, a habitat restoration plan. One I'm working on right now, which you'll get a chance to look at down the road, is a strategy for financing the CCMP. And those become kind of more of the tactical support documents where the rubber hits the road. So the work you're going to do today is really important because it's going to do a couple things beyond just the who, what, where, when, and why. This document, in conjunction with the CCMP, will be the documentation that I take forward as the executive director when we're making that case to, local, state, and federal policymakers and funding agencies to make the case. Why is monitoring important? The fact that we've got a plan for moving forward is going to be important to that, but also maintaining and hopefully expanding funding for monitoring at all levels. And we have been moving that forward, but I can tell each of you that one of the things I hear most often, whether I'm in Tallahassee or Washington, is often, nobody's out there, watching the shop, or we don't need any more science because we just need to do shovel-ready turn dirt projects. And as Dennis said, what we know already from monitoring is that this is a complicated system. We know we have gaps going in. You're going to help identify those gaps. And so we will move forward with this document, and Dennis mentioned it, but once the draft document is in place, we will do an internal peer review through the management conference advisory committees. Once we are comfortable that we have kind of a science-ready consensus among the management conference, then we will move this document to a consensus, a formal consensus review with EPA. And then down the line in 2021 at one of our quarterly meetings, we will bring this document through the management conference again for recommendation for adoption, and then ultimately IRL Council Board of Directors adoption. So with that, Dennis, I'm going to just stop and say thank you all again for your time. I'm going to be popping in and out with another Zoom meeting that's going on with the EPA right now, but I wish you well in your efforts today, and this is important work we're about to do. So I think next up in the agenda is Chuck Jacoby.
So thank you, Dwayne. And I bet Chuck is surprised that both Dwayne and myself have given our talks, and he has extra time going into his talk. I'm sure that nobody would have expected that between the two of us. And I just want to thank Dwayne for his leadership. I don't know what the NEP would be today without him. So thank you, Dwayne. Okay, Chuck is going to lead off the first of 3 sets of talks. And he's going to talk about the One Lagoon water quality component, what's going on now with monitoring with water quality. You're up, Chuck.
Okay, so with any luck, you're seeing a PowerPoint slide that says water quality. Looks good. Okay. So first off and foremost, you've heard this is an interactive process. So please don't shoot me or the other messengers. This is an effort to kind of feed back to the people who had input what we received or interpreted. And I think everybody's happy to adjust what we have as needs be. My approach in this particular effort has been to parse the information kind of according to the list you see on the slide. Why first, who, where, when, what. Talk a little bit about access as we go along, and then right at the end actually kind of wrap up what we think these efforts are used for in terms of management in particular. So I'll be talking about the water quality part of this vital signs wheel. There are 7 items that are listed in that section, 5 of which we'll go through explicitly, as it were, and 2, legacy loads and wastewater, are more implicit in what we're talking about today. So, but to some extent, they're all touched on with the programs that we've seen so far. So overall, there are 21 programs, 15 of which predominantly seem to address stormwater or impaired waters, 1 that addresses hydrology, specifically an atmospheric deposition station, and 4 programs that look at contaminants, and particularly in sediments generally. The map on the right there is the kind of map that you'll see as you go through. In this case, that's all in, except 1 program, which is the EPA's coastal condition program, which is 5 random stations each 5 years, and we weren't quite sure what to do with random stations. So they don't get on the maps, but they're in the mix. This is the kind of thing that you'll see as we walk through. So there's a map there that'll show you the station or stations. Also try at some point to give everybody an idea of the period of record, what kind of access you have to the data, the frequency of measurements, the parameters that are measured. As I said before, the use will be summarized mostly at the end. So this atmospheric deposition station, it has over 10 years of data, although there are some gaps where we did not have funding. The data, you'd have to request the data. They're not available readily online or anywhere. There are continuous meteorological measures and some operational measures. So you can see the list there. The operational ones are things like the airflow through the filter for dry deposition. About weekly, and so we're doing wet and dry deposition. About weekly, things go into the lab for analysis. So filters are pulled and sent. If there's enough rainfall, a wet deposition sample will go to the lab, where the focus is on, the various constituents that you find in the deposition. So that's listed at the bottom. There are 14 hydrologic stations operated by USGS. They provide essentially what's continuous data on stage and discharge, greater than 10-year record, although again, that varies by station. Sometimes they come and go a little bit. And these data are available online, so you have access to those. There are 4 programs that focus in predominantly on contaminants and things in sediments. As far as I know, these data all have to be requested. I don't think there's a repository for them that you can reach easily. We'll go through the EPA program first, which again is the 5 random stations each 5 years. They've been going for 10 years or more. They do a fair bit of looking at a variety of things, the organic content of the sediment, grain size, organic chemicals, metals, toxicity tests, and they also look at fish tissue as part of what they do. The other programs that fall in this category, the National Park Service does 30 stations each 5 years for predominantly sediments. It's a program that's been going for over 10 years. Organic content, again, grain size, organic chemicals and metals in sediments. And those are the light blue triangles up near the northern part of the system. We put the NASA work with alligators near the Kennedy Space Center. We're not quite sure exactly where the alligators always come from, but again, over 10 years of looking at contaminants in alligator tissue. There's Mussel Wash Station down in around Sebastian, and they again has been going for a while. They look at sediment and oysters, looking for or testing for about 250 different contaminants and also bacteria in sediment. There are 15 stormwater / impaired water programs. So water sampling water has been a big focus for a while. For example, our program goes from 1996 onward here at the St. John's District. All told, we tallied up 611 stations, 567 of which have a fairly long period of record. There are 35 stations that provide what I'll call continuous data, which is hourly, or in some cases potentially shorter, and 576 stations that provide what I'll call discrete data. So that's typically monthly time step, weekly maybe in some cases, monthly or longer. So we'll look at the continuous data first. And so here you see, again, these are the continuous stations in the map run by different organizations. So there's a batch of different programs there. These collect, I guess you'd call them your standard physical and chemical parameters, shown in the yellow. And in some cases, a few extra things like oxygen reduction potential, partial pressure of carbon dioxide, and fluorescent dissolved organic matter. Some, relatively few, couple of programs have nitrate and phosphate that they measure on a continuous basis. A variety of pigments, mostly chlorophyll, but also others that are measured as well. So that's your biological estimates of phytoplankton abundance. Water movement in some cases, current, direction, speed, or discharge. And then the Harbor Branch stations in some cases have MET data. So if you look at the access for these data, when you go, there are 3 programs out of the group that have their data online. And so you can see the red arrows kind of point out the ones that you lose. So if you squint at those 2 maps, you can see what disappears when you try to look at online data. Of those online data access, there are 2 where you can actually download the data. I won't venture to speculate on the user friendliness. I have used ours for sure. You can get there if you're persistent. In terms of the sort of more traditional discrete programs, there are 9 that have been operating. And just kind of give you an idea, so I have been approached at times where, you show a map like this, and then because of the shape of the lagoon and so forth, if you get symbols big enough to see, it looks like boats are running into each other on a continual basis out there. But if you look, say, at the St. John's program, which has about 43 stations done on a monthly basis, and we pull like 4.5 liters of water. And so if you kind of multiply that all out, that's like about 2,300 liters of water per year, which is about 0.2% of the water that's in the lagoon. So that's what you're basing your management decisions on, that kind of effort. There are some of these programs, there are 2 programs that basically focus on field parameters, and those data are available by request. There are 7 programs that do field and lab parameters. 5 of those are uploaded to database of some type that you can access. Once again, the user friendliness may be in question. These are the kinds of field parameters that are measured by the different programs. So some of the things about overall conditions, weather conditions, a variety of a couple of metadata, things like depth of collection and water depth, variety of the standard physical chemical measurements in yellow there, factors that affect light penetration, which are highlighted in green here, and then something about the light regime itself, whether it be Secchi depth or taking measurements to calculate an attenuation coefficient. The laboratory parameter list is a bit longer. So again, a variety of physical chemical measurements in the sort of tan and yellow, various forms of nutrients, nitrogen, phosphorus, silicon, and carbon, all in the blue there. The biological measurements mostly focus on photosynthetic pigments. Major and minor metals, some of those would be, sort of micronutrients as it were, and others might be thought of as contaminants as well in the pink. Batch of other ions in some cases that DEP does. And then the two programs, the DEP program and the FDEX programs, also focus in on bacterial contamination. So when we looked at these discrete programs and the information we had available, looked like most had a rigorous sort of post-collection QA/QC process. So the data are evaluated and coded as need be. When you look at those 7 programs that undergo that kind of QA/QC, the monthly time step is true of about 4 programs, and it's longer for the others. And it's the red oval at the bottom there highlights the fact that for South Florida, their water quality, it's part of their overall set of stations that goes monthly. The rest go 7 times a year. When we go through the use, so there were 7 programs where we didn't really have a good handle on how they were used to guide management. The Park Services sediment program is used, locally, which makes sense given their range of sampling stations. Moderate use, there were 6 programs where we could see that, and that in there I would also include that EPA program that we didn't map. So their 5-yearly look at contaminants. And then there are about 7 programs that have been used fairly extensively, and that includes the hydrology to get at loads. The moderate use also included the atmospheric deposition you might have noticed, which again also was used to develop loads. And there you go, Dennis. Did I make it?
Well, thank you, Chuck. And Chuck left a lot of time for questions. So again, please enter your questions in the chat box. Okay. Well, Chuck, I think that means that you are the expert and everyone believed everything you said 100%. There is a question. Vincent in Cameo says, is FDAX sampling graph samples? What is their frequency and how can their data be accessed?
I'd have to go back and pull out the spreadsheet. So let me get back to you.
Yeah, actually, Chuck, David can answer that. So again, David did a lot of the legwork. So
You did all the
mute myself and David.
Hi, everybody. I actually used to work for this program years ago. Those bacteria samples are actually used for shellfish management. They're grab samples, they're permanent station locations. If an area is actively harvested, those grab samples are at least once a month. For areas that harvesting is not allowed, those samples can be, as little as once every 4 months or so. All right, we've got question for Chuck. Any big picture surprises when you looked at the data?
I think there's a FDEP question in there that we might have skipped over, and that goes all with their whole status and their sort of trends efforts. So it would go into your TMDL processing. If you look at when they do their impaired waters runs, where they take all their data and run it through to see what, where impairments have appeared or, where things are improving. That would be their bacterial work, Lisa. What big picture surprises when I looked at the data? I mean, I haven't, in terms of the distribution of things and what is measured, I guess no real surprises, potentially because I bump into it a lot. But are there gaps or things that we might want to improve? Yeah, for sure. And I think we should talk about that. I think one of the biggest things for me is the pendulum of how this is going to get done is probably swinging again. So early on, it was kind of pieced together with a batch of different organizations. It tended to get centralized as people became a little concerned about quality control and quality assurance. But now resources are, I think, pushing us back the other way again, where we're going to have to partner up to make this happen. Otherwise, we may not be able to keep up with what we think we want to have. How representative is one atmospheric deposition station? And yes, there was one at the Space Center that is no longer active, and it would definitely be worth probing into those data to see what sort of differences we saw. I think the answer to how representative, it probably depends on the question that you're asking. So if we're trying to parse out deposition over, again, a fairly long stretch of the coastline, it may not be adequate.
Does anybody else have questions? Okay. If not, you can still come in if you want. So we're going to try to do our first survey. And so we worked on this a couple of different ways. And we're going to have one in each of the next 3 sessions, counting this one. We're going to give everybody 8 minutes. We're going to send the link in the chat now. Wyatt will send it to you. And you'll see that in each time we do this, the first one asks you to rank a bunch of things in a particular category. And then most of the rest of our questions is to try to get a sense of things like, do you think we're sampling temporally and spatially in an adequate way or not? And then lastly, to what extent you learned new things today, how much did you know before, how much is new, that kind of thing. In all cases, you can put short answers as well. We encourage that. And at the very bottom, you can put anything else you want to tell us that's on the general topic of water quality monitoring in the lagoon. And again, we will show you some quick results later, but the fill in the blanks where there's going to be a lot more thought involved, that will come later, and we'll send that to you at a later date. So 8 minutes. If you have any questions, just send them in the chat and Wyatt can answer. Okay, so we gave everybody more than 8 minutes. There's still a bunch of people doing it. we're gonna just give you a couple more. But meanwhile, Jeff, if you could be ready to put your screen up, get all set, that would save some time. We're actually gonna let the poll stay open. About 5 minutes more anyway. So if you still need more time, we encourage you just to finish the survey. And Jeff, are you there? Sound wise?
Yes, can you hear me?
Yeah, I can hear you.
Great.
So while we're waiting for a couple more people to finish the poll, so I think everybody pretty much probably knows you changed jobs, but maybe not. You want to tell everybody about your job change?
Sure. So after 15 years with FWC, I moved on to Ducks Unlimited. Ducks Unlimited has been around since the 30s as advocates for not only waterfowl, but wetlands in general. And so I'm the first regional biologist for the state of Florida. And we're working on wetlands and associated habitats, waterfowl, and associated wildlife.
Jeff, go for it. You can tell us about One Lagoon habitats.
Great. Thanks,
Well,
Dennis.
we will keep the survey open for another 5 minutes and then we'll shut it down.
Sounds good. Thanks, Dennis. Afternoon, everyone. Hope we're all safe and healthy these days. And speaking of healthy, today we're going to be talking about the habitats of the IRL. IRL is quite famous for its habitats, and we all know that species require them. We cannot have species without healthy habitats. So it's a primary focus of the CCNP. General categories for the IRL we're going to be talking about today are submerged aquatic vegetation, coastal wetlands, and filter feeders. Habitats themselves are listed as vital signs, but they also touch on numerous other vital signs, all the living resources one's species of concern, everything to do with water. Good water quality, of course, is important to habitats. It also touches on harmful algal blooms as possibly affecting them as well. And when we talk about monitoring, of course, we have to think of it in terms of spatial and temporal components. And so for the purposes of the talk, you look at the graphic there, I'll be looking at this in terms of the spatial components. And this is taken from the Nichols paper on seagrass monitoring. It's very useful for this discussion. So we'll be looking at things from the landscape scale, the very large scale, the ecosystem scale, so the medium, and then the patch and organism scale. And we refer to those as tiers from tier 1 to 3. In terms of submerged aquatic vegetation, that's in reference to seagrass beds, macroalgae, in terms of attached and drift in the lagoon. Macroalgae is captured in a lot of the programs you're about to see. There's also some work that's been done with sonar that we can see as a sort of a baseline. It was limited in space and time. But it doesn't include the connected waters, so things like balasinaria and other submerged things in our tributaries. So the purpose of SAV, seagrass mostly, and with some macroalgae, lagoon-wide, we have 4 lead partners and 4 programs. It's a partnership with the water management districts and lots of their friends that work on mapping and fixed transects. Kennedy Space Center also has long-term monitoring. South Florida Water Management District with partners has a specific program looking at SERP recover, so the Everglades restoration and the monitoring of it. That's programs referred to as NESI. And then Smithsonian, coupled with aquatic reserves with the marine geo sampling. So let's look at these a little more closely. We're looking at the broadest scale, the tier 1 scale. It's imagery mapping that happens every 2 to 3 years since 1999. This happens with Water Management District partnering. There's some ground truthing that goes on. It's a gross scale mapping of persistent versus fluctuating. as you can see on the map there that's produced. It's lagoon-wide, but we need to remember that that doesn't include the Halifax, which is part of the NEP boundary. So when I say the term lagoon-wide, I mean not including the Halifax. At the Tier 2 scale, it's the South Florida Water Management District NESI program that is capturing data at this scale. As you can see, it's a tessellated mapping set up to do randomized sampling. Southern end of the lagoon is divided into segments, and then there are quadrats within those segments that are sampled. At the patch scale, Tier 3 scale, first we start with the Water Management District Partnership. The fixed transects have been going on twice a year, established in the early 90s, and some sites added later, lagoon-wide. Kennedy Space Center, also quadrats long fixed transects that started in the 80s. That's concentrated in the southern Mosquito Lagoon of Banana River. And with the NESI program, quadrats long fixed transects, and you can see there's 3 short parallel transects in each of those cases. 4 sites started recently in the southern end of the IRL. And then lastly, the Smithsonian work, also quadrats long fixed transects. A few sites done starting a few years ago in Indian River and St. Lucie counties. So we look at the spread of these across the lagoon, and some mappings coming up soon. Just remember that there's some common methodologies here. You heard the common theme of quadrats along fixed transects, things like seagrass cover or algal cover, density, blade lengths of seagrasses. So lots of commonalities in the parameters, metrics that are measured. And the way that a lot of this is analyzed, especially for Water Management District ones, that could be considered at the tier 2 level, given the spatial and temporal component of it all. But according to the Nichols paper, by definition, it's a tier 3 monitoring. A very comprehensive program. So the differences among the samplings, the quadrat size can differ, and the frequency of sampling can differ. So starting at the north end of the lagoon, the Water Management District transects are shown there in red, very comprehensive. The Kennedy Space Center ones that are shown in black gives you an idea of the spread of those. Moving to the central lagoon, we continue with the Water Management District ones, and there around Fort Pierce Inlet, we start to pick up Smithsonian and the South Florida Surf Recover transects. And then moving to the southern end of the lagoon, more of the same. As you can see, Surf Recover, Smithsonian, as well as the Water Management District ones. In terms of filter feeders, general categories here: oyster reefs, clam beds, and other. And you'll see those on the slide there. In terms of clams and other, there's really no real monitoring going on. Have been some scientific research projects that were limited in space and time and scope. So for the purpose of this talk, we'll be talking about oyster reefs specifically. And within the IRL, we have 5 lead partners and 3 programs. Firstly, being a partnership of FWC and South Florida Water, again associated with Recover Everglades restoration monitoring. Florida Oceanographic Society has an oyster spat monitoring program. And then lastly, Smithsonian, as part of their Marine Geo program that we looked at with seagrasses, also includes oysters. So as you look at the Tier 1 scale, good news on this is most of the mapping has been done within the lagoon, mapping of oyster reefs. However, it's been sporadic over time. Some of them were done as recently as 2018. And a good repository for these sorts of mapping data are at FWI repository, and I'll show you that link at the end of the talk here. But some of this involves ground truthing. The work that was done in 2018, for example, used live cover as a boundary, because our concern here is that some of these beds are partially dead or completely dead at times. And so looking from this tier doesn't tell you the whole story. There's nothing happening at the Tier 2 level, to my knowledge. For Tier 3, monitoring is happening at the patch scale. This is the work that's ongoing with FWC with regard to Everglades restoration monitoring. That sampled 9 stations periodically, St. Lucie and Loxahatchee specifically. For Florida Oceanographic Society, that's 6 stations sampled monthly, and that's restricted to the southern end of the lagoon, including the St. Lucie And with Smithsonian, we're back to the stations we mentioned before in the Indian River, St. Lucie County area. It was put together just recently. Let's look at these in mapping. We have to start with a central IRL, so nothing happening to the north, concentrated around Fort Pierce and Vero. And then as we look at the southern IRL, we start to pick up the other stations, including those from Smithsonian, mostly centered around the St. Lucie River. The main concern there, of course, being surf recover. So, as I mentioned several times here, most of what we do in the way of filter feeding is at the southern end of the lagoon, and it's related to major water issues, water management, and recover for Everglades restoration. But we could add to that a really interesting study that was done by FWRI, in which they looked at those extensive reefs at the mouth of the Sebastian River and looked at the loss and gain of those reefs over time. So that serves as a good baseline. We could look into that further in the future. So in terms of coastal wetlands, we're referring to salt marshes, mangroves, living shorelines, spoil islands, and the floodplain marshes and forests of the connected water tributary rivers. In this category, we've got 4 lead partners and 4 programs, the water management districts, working together, doing land use mapping and tracking changes over time. St. John's also working with National Park Service on looking at the elevation changes within certain marshes. It's referred to as the SET program. And then lastly, UCF, Water Management District, some counties, FWC also partnering, working on characterization of the shoreline of the Indian River Lagoon, looking for possibilities for us to do living shoreline work. So as you look at the Tier 1 scale, this is the aerial imagery mapping that the water management districts have done for St. John's. It's analyzed every 5 years since the mid-90s. It's lagoon-wide. And very helpful, as you can see, it shows us the changes. You might be able to see some changes from the 20s to the 90s there. So very useful at this large scale to show us large-scale changes in our coastal wetlands. And those maps can be used for reports like this one. So this is the coastal habitat mapping report that FWC put together years ago with lots of help from their friends. So we look at the north end of the lagoon. So this is sort of the existing extent of our coastal wetlands. And we can look at the southern end of the lagoon as well. And then at this sort of Tier 3 level, that's in reference to the UCF Water Management District partnership here, looking at shoreline characterization. They evaluate the shoreline every 30 meters, starting with this aerial view first. If it's natural, it's every 100 to 300 meters, and they take various parameters, various metrics that are measured. So we have this baseline that was captured for us recently in Volusia and Brevard. Other counties, those are going to be done now or soon. So it provides a good baseline for us moving forward. We could use this as a monitoring effort. And then very much at the PAT scale, Tier 3 scale, we look at wetland elevations. This is the SET program that's done. It's an interesting little apparatus so they can get measurements down to the millimeter of changes in elevation. And the frequency varies for these sites, but it's only done in the North Lagoon. So here's a map of those sites. So Mosquito Lagoon and the very far reaches of the North Lagoon. We have some of those SET stations happening. What's the point of all this? Well, all of the products you saw there are very useful. Some of the reports that have been done are the NNPs that FWC put together with partners from around the state. The first one is S is seagrass, the next one is CH, coastal habitats, in other words, wetlands. And the next one, the O stands for oysters. So those are very useful documents. A lot of the mapping that's done are captured within there. Speaking of mapping, the FWC repository there, link that's shown, is a good spot to find a lot of these maps. Most of the other data you would have seen, however, is by request only. Water Management District also has some good mapping features on their websites. But these are all used for trend analysis to be able to predict and form adaptive management strategies. Obviously, all this habitat is strong tightly to water quality, and so when we couple water quality with the habitats, then we're able to work on things like models, and make predictions and so forth. And all of this, of course, leading toward target determination and our ability to determine success of restoration. And that's it for me. Back to you, Dennis.
Okay, so if there's questions, please type them in the chat. And Jeff, if you are able to keep up with, sometimes, the chat scrolls and we lose track of where we are, but I think so far, you could probably, if anything comes up, you could probably just read them yourself. If you need help, we'll jump in.
Very good. Can you briefly explain the importance of wetland elevation? Sure. Wetlands do, in fact, usually change over time. They, for example, keep up with sea level rise. And there's some really interesting studies where they've done coring of the sediments, including in Florida, to see how wetlands have kept up with sea level rise even in the last 5,000 years or so. One of those reasons is to look at things like sea level rise and those changes. The ability of marshes to keep up with sea level rise, is an important one. But you can also consider places like Louisiana, where they've cut off the sediment loading that's coming down the Mississippi River and feeding those marshes. So they figure they lose about a football field a day of marshes. And so it's really important that sediment continue to get into these marshes so that they can continue to build over time. So it's a good thing to track.
Okay, if you have any questions, you can still go ahead and put them in. But meanwhile, we're going to queue up the next survey of the participants. And you'll see a format very similar. The first question has kind of changed a little bit, but most of it is very similar, just a different topic. Habitats.
Okay, another question. What do you think the biggest challenge will be moving forward with monitoring habitats? Well, I think we've done a tremendous job with seagrasses, but with all the other habitats, we're really struggling there. So I think it's a matter of having, the local will, the political will, and the funding that will need to be done. Coastal habitats, wetlands, in other words, the aerial view is important, but the on the ground, I think, is equally important, and that's something we're definitely missing. Species composition and density, canopy height, and all those things. We did have a major die-off of mangroves as a result of Hurricane Irma in a 50-acre impoundment in Jensen Beach, and we just wonder if that's the future for us. So we really have to be monitoring these things more closely in preparation for potential stochastic loss.
So unless there's questions, the survey link's been up there. You can go ahead and start anytime, of course. We'll start with about 8 minutes and see how we're doing.
Dennis, I didn't see an option when I was going to share for PowerPoint application. Has everyone just been sharing their whole screen, or is? Let's see, application.
Yeah, Wyatt's going to jump in.
Oh, now it's there. So are y'all seeing that now?
Yeah, it looks good here. Okay, so it's 2:30, time for Derek. We will keep the survey going for another 5 minutes, although it looks like just about everybody who did the first, same numbers have almost completed the second survey. So without further ado, Derek's going to talk to us about living resources.
All right. Hey, everyone. For those of you who don't know me, my name's Derek Cox. I'm with FWC's Division of Marine Fisheries Management. and I'm going to be presenting the living resources monitoring programs that we have in place so far. So there's over 4,000 species that live within the Indian River Lagoon system, and 53 of those are threatened or endangered. So it's really important that we monitor certain vital signs within these living resources to ensure conservation and balance of these species towards this, ultimate end goal of having a healthy lagoon system. There are 29 monitoring programs covering all six vital signs from living resources. 5 of those where those monitoring programs are focused on those specific vital signs, and then 1 with the dotted or dashed red line there, exotic and invasive, that's addressed kind of indirectly. It's not the focus of any specific monitoring, but it does get incorporated into some of those other ones. Let's see. So within biodiversity, we have 3 monitoring programs currently. Species of concern is by far the largest here with 16 programs in place. Fisheries and forage fishes, 4 different programs, and then harmful algal blooms, we've got 6. As I go through this presentation, I'll kind of describe each of these monitoring programs within each vital sign, kind of their geographic extent and the frequency that they're sampling. But kind of keep in mind that these projects all have different objectives or side questions that they may be answering, so there's a lot of overlap. They don't necessarily fit into one category and exclusively one category as some of the other monitoring program descriptions earlier also did. So we kind of placed them into where we felt they fit best, but there definitely is it's not always black and white as to where they're monitoring. Just to orient you a little bit for how these slides are generally going to be set up, the vital sign will be up top there, and then on the top right-hand corner, you'll have that table, which has the 6 kind of areas within the IRL system. So Halifax River, Mosquito Lagoon, Banana River, North, Central, and South IRL, with some X's just marking where that monitoring is taking place. Below that, you'll see if there are stations, how many there are, and then the frequency that sampling is occurring. And if there's a map available, it'll be below that table there. So we'll start off with biodiversity. So you have the Smithsonian Marine Station doing some benthic and faunal monitoring that's been taking place since 2005. This is looking at community composition, abundance and diversity of these infaunal organisms, along with sediment characteristics, and then taking water parameters as well. You'll see a lot of these living resources monitoring programs are set up to take habitat and water parameters along with where they're finding these living resources. Also, Smithsonian Marine Station has, as part of their Marine Geo program since 2017, has been looking at fouling species within the central IRL. There are also, just coincidentally, 15 stations here, similar to the last slide, and this takes place every year. So they have 5 habitats: oysters, mangroves, seagrass, stocks, and soft sediment, and 3 locations. You can see on the map there all the different colors and shape symbols represent the different types of habitats. And so they put these fouling mats out for 3 months and see which organisms start to develop on them. So things like sponges and tunicates and things of that nature. And these are also very helpful with invasive species. Again, kind of not focusing on that, but kind of a nice byproduct of these is that we get to see a lot of these invasive species as they start to invade the lagoon, as a lot of them are members of the fouling community. The last one here with Smithsonian Marine Station is kind of more of an overarching diversity and species inventory that they started last year. So this is really just looking at kind of species occurrence and then status within the Indian River Lagoon system. And so these are kind of opportunistic sampling as they find, either new species or new occurrences of species to get collections of those and eventually to sequence that DNA and make barcodes for each of these organisms, as well as looking at things like diversity within organisms as well. Moving on to species of concern. Excuse me. Our first one we'll talk about are manatees. So FWC has been doing aerial surveys for abundance and distribution of manatees statewide, but obviously including the Indian River Lagoon there since 1985/2012, depending on the methodology. Along with this, they are going to be taking temperature data at those stations in the IRL. In the map, you can see there kind of pretty good coverage north to south, and then also monitoring health of these manatees that they are surveying. Kennedy Space Center and USGS also have monitoring programs in place for manatees since 1977. Sorry. Kennedy Space Center has also been doing aerial surveys for abundance and distribution, also recording environmental conditions. These surveys have been twice a month during the summer, so June through August, and then monthly for the rest of the year, this being kind of more near Kennedy Space Center, so Northern IRL, Banana River, and Mosquito Lagoon. And then USGS since 2011 has been doing some GPS tracking of individual manatees with their movement, habitat selection, taking health parameters, and as well as doing some discrete sampling every 2 to 3 weeks in the winter. Bottlenose dolphin, we have Hub SeaWorld Research Institute since 1999, doing mostly stranding response, but also photo identification in the Halifax River area. And this is taking place most areas except for the southern Indian River Lagoon. And they're also looking at things like distribution and health parameters for these monitors for these dolphins, when they are responding to these strandings. FAU at Harbor Branch this year started looking in the central IRL at dolphins and doing some photo identification as well, and also looking at behavior, movement, calving, and foraging within this area. Diamondback terrapin, we have a citizen reporting science monitoring program that the Brevard Zoo is coordinating since 2013. So this is opportunistic reporting of diamondback terrapin throughout the IRL system. And sea turtles, we've got the UCF Marine Turtle Research Group since 1982 in the central IRL using this entangling net bi-monthly to kind of catch turtles and look at things like health, movement, demographics, diet, and diversity as well. Birds, there are a lot of different monitoring programs going on for. I'm sorry. Oh, okay. So with birds, we've got typically it's looking at wading birds or sea shorebirds, and so a variety of players in this: FWC, Kennedy Space Center, Martin County, and DEP aquatic preserves. And so most of these are looking at nesting attempts and successes. There's not really stations, it's more kind of regions or areas, but these are taking place throughout the lagoon as a whole, and most of them are happening monthly. So looking at, like I said, a lot of nesting attempts and successes, population distribution and estimates, and then habitat use and site fidelity, depending on the program there. Horseshoe crabs have been monitored by FWC since 2018. You can see on the map there the stations that they're going to be looking at spawning density and taking samples from April through June. And then river otters are, again, a citizen reporting to get abundance and distribution, in addition to some camera traps that are set up to help get further information. And then also looking at health and mortality since 2016. Fisheries and forage fish I kind of combined into these two slides since a lot of them overlap, but we have our fishery dependent monitoring program that FWC has been in place since 1997. This looks at both recreational and commercial data for things like catch and harvest and effort of typically more targeted species, and it's also been going to be collecting biological data, things like otoliths, mercury content, potentially a variety of other things that they can look at kind of opportunistically depending on the species. We have some fish abundance and movement monitoring programs as well. We have FAU Harbor Branch since 2016 has been doing an elasma rank monitoring program where they look at CPUE throughout specifically, I believe the central IRL, but may be further than that. I have to double check that. But they are looking at elasmobranchs and kind of their distribution and movement. And they also have acoustic telemetry set up. So the FACT network, Florida Atlantic Coast Telemetry, since 2008 is what this map on the right is. And you can see all the stations color-coded by their the owners of those receivers. All these receivers can detect tagged fish if they're tagged with acoustic transmitters. And there's an agreement between all of these groups to kind of share data so we can get some really interesting information on fish movement throughout the entire IRL and the whole coast of the U.S. Fisheries independent monitoring, in place since 1990, has full coverage of the IRL, but is not at specific stations. It's more specific zones and random within those zones, which you can see on the map there. And this is going to be looking at relative abundance and diversity of fishes. This is where we get a lot of our information from juveniles, as well as through forage fish. And they're also going to be taking biological samples of these as well. And they get some really good water and habitat data that corresponds with where these fish are located. They do this through a variety of techniques, but a lot of them involve pulsings. We have the Fish Kill Hotline, which FWC has been operating since 1995. And this is going to be citizen-reported species abundance and diversity of various fish kill events. Our last vital sign here are harmful algal blooms. So these, again, are being monitored by a variety of groups: FWC, UF, FAU Harbor Branch, St. John's River Water Management District. You can see on the map there kind of the spread of these stations. They're going to be looking at things like abundance and composition, as well as taking water quality data. Some of these stations are set up to be, kind of just a general monitoring, I guess, where it would be, monthly or weekly, and then they ramp it up with additional monitoring stations and sampling if there is an event, a bloom. Similarly, phytoplankton and chlorophyll monitoring is taking place. NOAA, South Florida, and St. John's River Water Management Districts, and UF. Again, looking at species composition and biomass. These two maps here show on the left the St. Lucie River, kind of as water makes its way from Lake Okeechobee out the inlet. And then the map on the right shows the different zones that St. John's River Water Management District has, and the orange dots represent the phytoplankton monitoring stations there. And in addition to the composition and biomass, they're doing water quality monitoring and chlorophyll concentration in some of these. And so just to kind of wrap things up, there's a very large diversity of organisms within this living resources category and throughout the lagoon, and that kind of necessitates a high variability in these monitoring methods that are being used. These can be very species dependent and often require unique strategies and approaches rather than kind of a one-size-fits-all, this is how we're going to monitor all species within the entire lagoon approach. And it's really vital that we focus on these vital signs and have these monitoring programs in place for these key species. We can't monitor all of them, obviously, but if we select certain ones that we think are most important and make sure we are monitoring those, it can really go a long way for understanding interactions within the lagoon and the health of the lagoon. Like I said earlier, a lot of these things don't fit in their one box. They overlap a lot. Species of concern overlap with other living resources, and they all have interactions and dependence on the other categories as well, like water quality and habitat. So we really need to monitor these, keeping in mind their complex relationships with each other, so that we can be sure things are in balance and that we have conservation and longevity throughout the IRL for future generations. And with that, I will take any questions, keeping in mind that I don't actually do any of these monitoring programs, and so my knowledge is kind of limited, but I'll do the best I can.
Okay, so Derek, I think you got at least one question from Virginia. I don't know if you can see that or not in the chat, but it says, how will we how are HAB monitoring sites selected? Do we think they provide a representative coverage?
So that's going to depend on who is doing the monitoring. I'd have to look into it a little bit further to see how each group selects their sites, but we can look into that and get back to you. I imagine some of them are kind of opportunistic, where we already have something in place or where it would be easy to sample or important to sample, and then others are probably based on other factors, but I can look into that and we can get back to you on that. Do we have representative coverage? I can't easily pull that map back up, but I would maybe say what do you think to that? And that's part of what our surveys are trying to gauge is, do you think that we have representative coverage throughout the IRL system? There's definitely coverage definitely varies. There are more standard monitoring where it's happening, every week or month, and then when there's a bloom, there's increased coverage in that area that's affected. So it kind of depends. But that's one of the things that we're trying to kind of decide through this workshop here.
So Malcolm, can you hear me okay? Malcolm is here at Harbor Branch, and he is a PI for one of the HAB monitoring programs, and he's on the call. So I think, Malcolm, you probably have a pretty idea of the HAB monitoring that's being done by you, but also UF and everybody, really. Do you think you could weigh in on that topic of how the sites were selected? I know it's for different reasons by the different projects. And what do you think about representative coverage and how challenging, it is to monitor HABs?
Yeah, can you hear me okay?
Yep.
Okay. Yeah, what Derek said, I mean, the locations of sites are, to some degree chosen by what other monitoring may be going on. In my case, personally, the Irlon sites obviously are a great resource that we try to match up our sampling with. But coverage is a big, difficult issue. We can have very localized harmful blooms. Just this past summer, for example, there was one in Bethel Creek that was very intense and caused a big mess, but it was just isolated to that very small cove. So it's very difficult to monitor for all those types of events. So there's very much a trade-off in the work required to get adequate coverage and what we would really like to have. You can never really have too much sampling.
I think there's been some other comments made about the spatial coverage in the chat, but I think, Malcolm, don't you think too that the issue of how often we need to sample for HABs may actually be more challenging given the fact that, unlike, say, seagrasses, microalgae have such a fast generation time, they turn over very fast, the currents move very quickly. I don't know if you or Derek or anybody else wants to talk about that a little bit.
Yeah, temporal frequency rather is also a very big issue. Blooms can come up within a matter of a couple days. They can last for only a few days, or like what's happening in the northern IRL right now, they can last for months and months, or even years in the case of something like Oreolunga. So we really want to have high frequency where possible and long duration. It is a very challenging aspect of the ecosystem to monitor effectively. Right now, we do something on the order of 2 weeks, but that is barely adequate for a lot of the changes we see over time.
And Rich Paperno is, I'm sure everybody's seen in the chat, but if not, he's weighed in on the FWC sampling, which is done on a weekly basis, which is nice for temporal samplage, the thing we just talked about, and also talked about the locations, including Mosquito Lagoon. So that information is there. So Derek, I don't know if this goes back to you or if anybody else wants to chime in, but there was also a question by Virginia about how are decisions made about when to perform toxicity testing? And I think anybody wants to talk about the whole issue of, yeah, it's one thing to measure that there's the organisms, but is that what's important, or is it toxicity? So anybody on the call, feel free to chat in, or Derek, I'll let you take it off.
Yeah, I don't know the answer to that question. So if anyone else does.
Malcolm, is that something you want to talk about, toxicity? I know it's messy.
Dennis,
Yep.
this is Val. Can I say something?
Absolutely, Val.
You know, with HABs, toxicity is of course critical and important, but as we know, with brown tide, it's not the only thing. So I don't know that you would only want to focus on toxicity. Any bloom can be detrimental, as we know.
Yeah, I would agree with that as well. Thanks, Val. That blooms can lead to anoxia, which can cause widespread fish kills. That's maybe the main reason we see big fish kills as a result of blooms. Also, they can shade seagrasses, which have wide-ranging ecosystem effects. So there's multiple ways that these things can be detrimental to the IRL health overall. But toxins are definitely important and something that may represent a direct threat to humans. So they are something we should keep an eye on, and but maybe in a more targeted way, depending on the species we see out there blooming.
Okay, if anybody else has anything else, you can go ahead and put it in the chat. Otherwise, we're pretty much right on time. That's really nice of you all to stay on time. We're going to put the next link up. And one thing I'll mention is that since we're going to go into a break, what we'll do is we'll just put the survey up, and then, take your time and then take your break. And then we will reconvene at 3:10. And one other comment is that there's, I think, about 3 folks that we only can see a phone number. And if you don't mind either sending David or myself an email or a chat, it'd be good to know who you are, because we do want to keep an accurate list of participants. So we want to make sure that you get credit for being here. So if you can let us know who you are, that would be great. Otherwise, let's do the survey. And again, you can continue the discussions in the chat, or just mics will stay open. And we'll take a break, come back at 3:10. Okay, so it's time to reconvene. Looks like the surveys are not coming in anymore. But if we'll leave it open, I guess we can leave it open for another couple minutes in case somebody had to take a break first. How's that? Okay, so hopefully everybody is returning back if they walked away from the screen. This is Dennis again. I'm going to try now to very quickly summarize a very, very draft at the present time set of recommendations. And we basically just took a look at, I would say this is a very high level look. And that's what I had talked about with Dwayne, was that initially, our major recommendations are going to be at a fairly high level. But it's really important in the report, but for us to really flesh these out and to give some very specific details. And I'll mention that a little bit again. And I would say, just from the feedback we're getting in on our surveys, there's a lot of really good nuggets of suggestions that you will see. Those of you who have sent those in, it's probably just about everybody. I sent some good ideas in. You can see how it would be very easy to take that and incorporate it. Anyway, if we had to group these things into just say 4 broad categories, and if you want just like the keywords in your abstract, I think they are resources, access, use of the data, and gaps in the data. So I'll flesh out each of the 4 with some more bullets. Again, it'll be a very brief set of bullets. So in resources, what we're talking about here is we believe that we need to work collaboratively to obtain and deploy resources needed to maintain, expand, or initiate long-term data collection programs to meet demands of managing the IRL. With such resources as communication, coordination, and collaboration among those involved in monitoring. Well, that's what we're doing today. I put up on the screen another effort, I guess 5 years ago already, where we did an initial data workshop, which in some ways led to a portion of what we're talking about today. We think that that is probably the most important thing, is to maintain what we're trying to do today, what we're going to do with putting this report together, which is basically, open the lines of communication a little bit more, try to encourage more collaborations and more coordination of our overall monitoring efforts. So this is, we hope, is the first step. Money. Money makes the world go round, right? Need a whole lot of money. What George Harrison said, a whole lot of spending money here. Dwayne mentioned that one of the things he's working on is actually a financial plan for the NEP. And I think we all realize that the monitoring efforts that are going on already take a significant amount of resources. And if we say, okay, we need to do this better, we have gaps, we need to do more, we need to go to more places, we need to sample more things and so on, where's all that money going to come in the midst of what we, I think, all recognize the next few years is going to be a very difficult financial time because of the pandemic that we are currently in the midst of. I think initially we had funding first because without money you can't do anything. But I would argue that without that first bullet, working together, the communication, the coordination and collaboration, the money is not worth going for, or will certainly not be spent as well as it could be. So I think really it's this, I think really if there's a sequence, it's that. Another great resource that we have, and an example of this would be the work that MRC does. Now, other agencies, other groups do that, other organizations do it too. But I think we all recognize MRC as having a very long-standing, very good volunteer program, including really a long record of water quality monitoring at a lot of locations. I think increasingly the use of volunteers, call it citizen science, if you will, is really an important asset that I think we can build on. Certainly, that's been the case in restoration work, such as all the great work that's going on with oysters. Next one was access. What does that mean? Work collaboratively to design and implement an array of approaches that provide access to data, analyses, and interpretations that meet demands of managing the IRL, such as providing consistent and widespread access to data as they are collected, reviewed, and accepted. And the idea here is perhaps we need to have a portal with links to all active programs through the NEP, or perhaps maybe better, because NEP can only do so much. But if we partner with the right organizations, and Socorro is an obvious choice. Dwayne has already talked to Socorro a lot about collaborations. For those of you who don't know Socorro, Socorro is basically the southeastern part of the International Observatory System. And I put just a picture from their data portal there that shows you in general where they operate. Again, like Chuck said, when you put a bunch of big bullets on a map that covers a lot of geography, it looks like everything covered. Actually, that's not the case. It just looks that way in the one slide. But it is kind of a one-stop shopping, especially for the offshore, nearshore coastal waters. More and more, they are getting into estuaries, which is a good thing. So possibly, we could collectively work where there could be an IRL section of the Socorro portal, and they have funding to keep it going and so on. Usually the maps are not sideways. I put this one sideways to make it fit, but that's the way it looks like if you stand on land and looked out to the ocean. This is one of the maps that David put together. But one of the things is that, as I said earlier, that storing and providing access to the maps, the information we're putting together, and also the science plan that Claudia is heading up, as well is going to have the task of putting together a lot of the information they are going to come up with in terms of the actual use of the data and what it means and the long-term trends. All of that needs to be made available, in a very easily accessible location. Another topic that we've talked about a little bit, not so much today, and I did say that, we're not really counting in our monitoring program things that are no longer being done. But yet, we also recognize the importance of legacy data, things that may have been done before we really had computers sitting on our desk, for example. And a lot of the people that have collected the data perhaps are retired or no longer with us or trending toward that direction. And all of us, so I'm going to show you something I should never have shown anyone before. This is one of my secret legacy data sites. You may recognize it. But, I know colleagues who have file boxes sitting in their garages, and every now and then they say, yeah, I've got to make this available somehow. So in all seriousness, I think it's something that we need to talk about, given the incredible changes that have occurred, say, in the careers of some of the folks here on the call today. But I alluded to how different 10 years has been. But let's talk about how different the lagoon is in 40 years. Listen to one of Grant Gilmore's talks, for example. Next topic, use of the data. We need to work collaboratively to ensure that relevant data are incorporated into the adaptive management of the IRL, such as evaluating the status of and any trends and key parameters relative to agreed metrics. This is something that I think Claudia's group will do a lot more of, but I think that we need to understand that we have to have a long-term monitoring station in order to provide the data that's going to continue to look at those trends once we decide what the metrics really should be. I think on the monitoring side, it would be good if part of our monitoring, especially when data are more readily available, can trigger an appropriate response. So for example, all of these continuous water monitoring sensors, somebody picks up a low oxygen level, one that's going to cause great stress, one that's going to cause fish kills like Brevard County experienced a couple of years ago. We should have an early warning system in place for that. We should have an early warning system in place for things like harmful algal blooms. If we could detect toxicity, that topic came up before about how important it is to understand toxicity and so on. I mean, these are all things that I think can be done in the future, and we need to think about them when we discuss our future monitoring plans. We need to evaluate the effects of projects designed to restore the lagoon. So in my own work, periodically, I say, one of the reasons to monitor is because if some agency says, well, we're spending a few $1 million to make the lagoon better at this particular site to improve its water quality, we need to know, is that true or not true? If it's true, great. Everybody gets a good attaboy, and let's replicate that elsewhere. But we should also know about our failures so we can learn from our mistakes and make them better. We can't do that unless we do adequate monitoring. We need to adapt policy, regulations, operations, or monitoring to increase the efficacy of efforts to restore the lagoon and efforts to document progress. So that's kind of a little redundant with some of the things I've said already, but I think it needs to be worth being stated. And I think previously, early on, I did say that it's really important that we have accountability and accessibility to the public. In the end, they are our biggest customers, if you want to look at it that way, our end users. And we also need to respect that there is the need to consider the diversity of our large public audience. Lastly, gaps in data. We need to work collaboratively to identify and fill gaps in the data needed to support management of the IRL, such as gaps in the parameters or metrics monitors related to priority questions asked by managers and the public. Now, this is one of the things that we try to address very strongly today in our surveys, and I'll talk about them in a few minutes. But I can tell you that we got quite a lot of suggestions on gaps. That's what we hope to get, and we did get some really nice suggestions that cover both spatial and temporal questions. So that's just basically the location question and the frequency and monitoring question that we addressed. And we didn't talk about this quite as much today, but we hinted at it several times, and I mentioned it in this presentation, is that we need to, really upgrade what we're doing with improved technology. We need to start to look a little bit more about how to better do just data management and data synthesis, and then how do we take that information and tell the proper stories, if you will, to both managers and to the public. I mean, we have to make sense of what we're doing and what it's achieving. So this would be anything from using satellites to a much greater capability than we have in the past. We know how readily they can point out blooms, but there's a lot of other features that they could be used for. Or we can talk at the molecular level, where there's a number of efforts in the lagoon, certainly elsewhere in the world too, but in the lagoon, there are now a number of people that are trying to use eDNA, which, if you don't know what that is basically, it's all of the DNA that's sloughed off, say, by when a fish swims by a little patch of water. And the idea is that you can get perhaps a better census of the overall population, the overall diversity, using such a molecular tool. I think it still has a lot of fine-tuning that needs to be done, but it's something that will be done, and we need to figure out how best to use that moving forward. Just a few other notes, things that we're not going to talk about so much today, but which we certainly will for the report. And that is that the CCNP talks about different timelines for doing things. So, for example, short term is typically 1 to 2 years. I'd like to think of that as like, well, of all the things that we've just said we might want to do, if that in fact is what we want to do, because we haven't decided that, I'm just throwing this out as a trial balloon today for everybody. I think this would be like, what is a low-hanging fruit? What can be done, with not an awful lot of time needed, and with not an awful lot of new budget that we're not going to have to do. So if we take the horizon of 2020, and what a year it's been, I'm not sure we ever would want to use 2020 as a baseline, but that's what we got. There in 4 years, I would say would be midterm. Seems like a long time right now in the middle of the COVID. But in 4 more years, that's another election cycle at the national level for a new president. So in 4 years, we should be able to do some major things. I would hope, for example, on the data side of things is one possibility. And then with more resources and more time, we should be able to, I think, really be able to say we really do have a Indian River Lagoon monitoring network that is set up to track and evaluate long-term trends. And, so I realize that this is a minimally probably a 10-year effort to enact some of the recommendations we might have. And that was pretty much the timeline that Dwayne with the CCNP and which was, recently re-approved or whatever the word is, and the science plans and so on. We're all kind of looking at that 2020 to 2030 horizon. So I think for each of these gaps that we talked about, we need to identify, prioritize, and provide specific recommendations for those gaps. And the input that you have put in today so far, either in the chat or in the survey responses, is going to be really good information that can be fed into the report, as can some additional thought, of course. I think one of the things we did think that going into this is that we may need to think a little bit more about living resources if we are fully capturing them when we talk about monitoring the health of the IRL. So with that, I think we can allow a little bit of time for questions and or discussion, however we'd like to call that. But I would say that it's fine to call it discussion. Okay. So you're going to do the chat. I don't know how to get back here. Let's see. I guess I got to get out of that. And where do I gotta go?
First, let me stop this.
Yeah. Okay, now I can see the chat. All right, anybody have any comments? And at this point, if you've got a major comment, just go ahead and unmute. We're going to be crazy now. Just unmute if you have a major statement. Feel free. Okay, does everybody think that that short list of things, which had actually was a short list with a bunch of bullets, do you think that's a good starting point for the steering committee to work on? Or do you think, is there something that is glaringly wrong, glaringly missing, or is it way too optimistic or not strong enough? Well, David, how should we record that? I think we got it right. I don't know. Okay. Well, I'm not really sure. I can tell you that that's just a trial balloon. Okay. No, I thought there's something came up. That's something else. Sorry. Okay. Thank you, Virginia. All right. Keep the thoughts going. I think everybody wants to know how the surveys turned out. So did I get an email here with the... Okay, let's see. I have not seen this yet. So here we go. All right, can we get me back going here? So I had started just a couple little slide template to get this going. And then the way the survey worked is we could track things as it was going along, obviously. And then Kristen very nicely did some cut and pasting, and she dumped some of the data into an Excel file, and we have some quick answers. Tell you what you all said today in our surveys. So you remember there were 3 of them. The first one is water quality. So can you see the mouse? Is the mouse? Yeah, you can see my mouse. It's really tiny, but my computer wants to restart yet again. I think wait one more hour. Okay. If you look at the top, that's the way we saw the output. So we asked everybody to do their own 10 rankings, and you can see that they are now ordered from 1 to 10. Again, you know, we're mostly scientists or people who know statistics. So one of the things is that, you know, we'll take a harder look at these. But as an initial thought, I said before I had any idea what the results were, well, let's just look at what the top 3 were, and I'll say here's the top 3. So the top 3, as you can see, the top one is eutrophication, nutrient loading, and actually among all of us, 44% of us had that as our number 1 choice. So you can see it was number 1 overall, which factored the entire 1 to 10 scale, but it was the one that, of course, was number 1. Followed by that was chlorophyll A, dissolved oxygen, and I think we all understand that those three are highly coupled, and that's the thing. You know, you can say these are 10 separate things, but you know some of them are so intertwined, and so I don't think we should be terribly surprised that the 3 of them came out together, and I would think that none of us should be terribly surprised that the concern about nutrient loading, eutrophication, in fact leads PAC. If we look at the question on do we have adequate geographical coverage, about half of us, a little bit more, said it's adequate. Let's see, am I reading this right? Only about 15% said it's not adequate. So there are some gaps, but overall, I mean, at least half of us find it adequate. If we look at adequate and definitely adequate, really, it's about 85%. Okay, so that's actually pretty interesting. I'm kind of surprised that it is that high. For data collection frequency, similarly, we have 67%, so about 2/3 of us think it's adequate. About 10% of us, 8%, think it's definitely adequate. And about a quarter think that it's not adequate. Okay, so that again is very useful. We asked the question kind of somewhat to evaluate us, to see what we were learning today. And the question was, basically how familiar are we with the projects that were summarized today? And about a third of us said most or all, and about 2/3, not quite, 62% said, we were familiar with some of them. And we have a very group of people who do know a lot about these projects, obviously, because we're involved with some of them. So it's just a tiny sliver that don't know that. Now, if this was a management group of people, just all management, or if this was the public, I think these numbers would be, really, really different. But that's basically a reflection of who's here. On habitat quality, the number one answer preference was seagrass, and that had a number very similar to number one in the previous one. And 45% of the people had seagrass rated first. The other two in the top tier of three was wetlands and filter feeders. So that's again very interesting. I don't think we should be surprised about seagrasses. I think the mantra has been for 30 years that seagrasses is really a habitat that really determines the health of the lagoon. I think we are understanding that is certainly important, but also that we need to have a better handle on a lot of these other habitats as well. In terms of the question about how well are we covering habitat in terms of geographical coverage, the majority of the folks went with adequate. Only 6% thought it was definitely adequate. Okay, so not many of us think it's the way we should be doing it totally. And about more than a third, so 36%, say it's not adequate. So this suggests to me that this is something that really needs to be looked at. Data collection frequency, about half say adequate. Those numbers are pretty similar. 36.4 again showed up again. And 12% said definitely adequate. So 36% again saying not adequate. So there's a certain number that basically suggests that this needs some more detail fleshed out. Again, when we look real quickly at the comments, because it's really hard to summarize all the comments, so people send 20 different things. In most cases, it's 20 different things. And then how familiar were you with this? Was 64% were familiar. Okay. Lastly, living resources. Number one was harmful algal blooms. And that was 50% of us put that first. Plankton. Now, there's this. I could see where this could be overlapping because phytoplankton are here, and obviously they cause harmful algal blooms. So that was number two, followed by the commercial and recreational fisheries. You can see with geographical coverage, it looks like 71% say adequate. Data collection frequency 60.7, and so on. And there seems to be some discussion about what the colors are. Do we have a problem there or not? No, we're good. Okay. So that is it in a very fast pace. I just looked at it really for the first time there. May have misspoken, hopefully not too much. And we will be looking at those in much greater detail. And again, all of the data will become available for anybody who wants to look at it. And we will be inviting more input, as I said earlier. I wanted to talk just briefly, about next steps. I think I've said this already. But just to recap, or if you weren't there at the very beginning, the goal now is to turn everything we've done, all the information we have so far, into a final report. Dwayne is anticipating that we'll be doing that during the first quarter of 2021, unless 2020 gets a no-cost extension. I mean, 2020 could go on forever, who knows? But hopefully 2021 will come, and we'll do it in the first quarter. And I would guess by the second quarter, it should be a pretty finished product. I would like to send a draft out after the steering committee is happy with it, to everybody here who participated in the workshop, like I said earlier. And then it would be posted on the NEP website for comment in a fashion, I think, probably similar to what was done for the CCMP. And at that point, that would be up to Dwayne and so on to figure out how exactly that would work and the timeline for all that. But we really want it to be, open and transparent as much as possible. We really do want it to be as much of a consensus as we possibly can get. And I just want to thank everybody for coming here today, for participating in the steering committee and the staff here that are participating in the room, and of course the NEP. And I'm going to leave the remaining time for any discussion points that anybody might have.
Oops, look at that.
Okay. All right. Any comments? And again, we can go open mic night. Open mic night. Any thoughts, comments?
So I'd just like to thank Dennis and David for keeping the wheels on this wagon, which isn't easy when I'm part of it. So.
Yeah, well, thank you, Jeff. What are your general thoughts, though? I mean, I had asked earlier, do you think that, I'll ask it bluntly. Do you all think we can actually do this? Not write a report. Come on. Anybody can write a report, but can we actually do a monitoring plan that will actually make a difference? I'll be my own devil's advocate. What do you all think? I teach sometimes too, so I do call on people, especially if I can't see them.
So Dennis, this is Val. So, as we saw, there are a lot of monitoring efforts, some overlapping, some, of course, mission driven by our various institutions. So getting monitoring going and perhaps tracking it better so that we have a better idea of how it all synergizes is not going to be hard. Filling in the gaps, which is going to require either more funding or change in something that somebody's already doing, may be harder, especially in the near term, because I don't foresee a ton of new funding coming, but maybe I'm being pessimistic there.
Well, let me ask you a question, Val. I'll be my own devil's advocate. I can't believe I'm doing this, but you think there's anything that is going on now that doesn't have to be done? Are we doing some things redundantly? Could we be doing what we're doing more efficiently?
Well, that's a real good question, right? Like, we'd almost have to look at every effort in more detail than we've done, see where there might be any overlap or anything like that. My general sense is there isn't. There may be some overlap, but that, things are spread out, at least for instance, the water quality stuff. That's spread out somewhat geographically, which is a good thing. So I think there's probably not a huge overlap there, but I don't know. You and David have dug into this probably more than anybody. I'm wondering what you think. Well,
I think it's premature because we have gotten an awful lot of information. I think the that phase is now over, and I think we're just beginning, really. You know, what you saw today, I think, was the results of just beginning the early analysis of it. So I don't think we really know. I would say, though, in most cases, I would think that what is being done probably needs to continue to be done, because there was a need. You know, I mean, it's kind of circular, but there was a need, and unless the needs have changed, I suspect that, you know, most of what's going on has been good. Virginia wrote in the chat box, having better information and access to what is available should improve its use by manager. So I think everybody knows Virginia and everything she's doing up in Brevard County. So, and she is a very good person, you know, as a manager for me to ask her the question, Virginia, what do you all really want? I mean, what do you really need? And how can we do a better job taking our monitoring data and making it at least available to you? Now, I understand there's availability, but there's also sometimes interpretation and so on. What do you need as a manager? I think you need to be unmuted, Virginia. Can you unmute her, Virginia?
I guess I heard it.
Okay. You're gonna get unmuted.
Part two of Curtis, which I'm wondering.
How about while we're working on Virginia's getting online with voice, Vincent, everybody knows Vincent too. He used to work at FOS, and now he's Sea Grant in Martin County. Does a lot of restoration work, has been a scientist, but also he's a manager too. So he said accessibility to the public and our interpretation of that to them is highly important in addressing a program's adequacy and value. So Vincent, how well do you think most of us do as scientists? And how can you and Sea Grant help us do it better as an extension person? Are we using Sea Grant extension enough in the lagoon?
Hey, can you hear me?
Yep.
Well, that second part, I'm not really sure if I can answer that well, but certainly, I'd say yes in general that that we can be helpful. Sea Grant can be helpful in helping get the message, get data or how it's interpreted to the public. But I'm not quite sure on the specifics of at this point in time.
What's the most effective way to get the information to the public? Just any type of general thing. What do you find helps?
Geez. I think some of the web-based resources are helpful. I missed a lot of this. I've been trying to put my daughter to take a nap for the last 3 hours. So, but I think some of the stuff,
you have a monitoring problem of your own, then, don't you?
Yeah, I think some of the stuff I saw. There's so much monitoring, and there's a lot of people that don't know about any of it. So, and if you don't know about it, well, how useful is it?
Yeah, I think personally, it's amazing how many people that I walk into in public life, they have absolutely no idea what's going on. It's like, and I spend a lot of time doing communication and education stuff, and a lot of people on this call do that, and it's kind of amazing that there's such a large untapped group of people out there who still don't have a clue what the lagoon is and what the issues are and what needs to be done. So yeah, it's kind of a challenge at times. Okay, Virginia, can you talk now? No?
Can you hear me now?
Yeah.
Okay. Yeah.
Sounds like that commercial.
I have dueling computers going because I have sound on one and I have picture on the other, and that was my issue. So, off the top of my head, I think the ongoing question for managing, trying to stop the excess pollution is, of course, the sources of the pollution, and so anything that helps to trace those sources back up the hill, and that includes a better understanding of how much of the nutrients in the lagoon are legacy load versus the new and ongoing excess loading. We have a couple different calculations of how much too much nitrogen and phosphorus we have in the water column, but we don't really, I don't think, have a good appreciation for how much of that needs to be removed, reduced, whatever, by upland controls versus if we could clean out the legacy load, how much more upland reduction would be needed. And then I'm also sort of fascinated by the idea of patterns in these harmful blooms that we've been experiencing, to the extent that they start in similar places and grow in similar places, then expand to certain segments of the lagoon where we have very high residence time, and they sit there and fester for way too long. So if we understood those patterns better, would we be able to come up with strategies to disrupt those patterns and avoid the time and space occupied by harmful blooms faster than we can achieve the nutrient load reductions that obviously need to occur? So I'm sure there's a lot more, but those were the first three things that came to mind.
So I see Linda Walters made a comment to everybody. Linda, of course, everybody knows her at UCF. I would add that some topics do not need ongoing monitoring, but rather short-term data collection, e.g. microplastics, aerial coverage of oysters, etc. So, Linda, how best to interface that type of short-term data collection with monitoring? I mean, how do you think we could do that?
Can you hear me, Dennis?
Yes.
I think I straddle some fine line of what we're calling monitoring and what we're calling research projects. If you did a project every 5 years or 10 years on microplastics covering the whole lagoon, I don't know if that would be listed as monitoring or research. But I don't think you need to monitor something like microplastics every month or even more frequently everywhere in the lagoon till the end of time. I think your saturation, your rarefaction curve will level out pretty quickly unless there's some major event. So there probably are some things, that's one example. Aerial coverage of oysters, I don't think you need to monitor that every year, but every 10 years would be really helpful. So I think there's some really good balance. We focused on the things that are monitored once a year, twice a year, every month at this meeting, but there are a lot of other things that could be monitored less, and we would gain a lot from the information. I think that's why microplastics ranked so low in the water quality, because people don't think it needs to be monitored that frequently. And I agree, but I think it's still really important information, and we have just begun to understand why we should even care about microplastics. We don't know that much about human health and microplastics. If we did, we'd probably rank them higher on the survey of how important they are to monitor.
So do you think in the report, one could flesh that out say kind of what you just said, microplastics are important because yada yada, and also there's not really a systematic monitoring program, if you want to use that word, or just, you know, like we could say it's the way you did, you know, data collection every X number of years, and say this is what we really should think about doing moving forward.
And there's probably lots of other things that I don't think about that would fit into that category of
Sure.
some larger space of time between doing concerted monitoring. Yeah, I think there's a lot that could be gained, and it would cost a lot less money.
Mm-hmm. And that may very well be true, because for other things, like as you know, we've kind of struggled with how better to talk about monitoring biodiversity, because biodiversity is actually pretty complicated. And, you know, with the stuff that we currently are looking at overall, it's not really all that much. The question is, what really should we be doing? But that's, again, the kind of thing where, you know, if it was done every 10 years, would that be adequate? You know, and just do it really well, and somehow get all of the people like us on this call and others to get together and just do it every 10 years. I'm just making that up on 10 years. I don't know if 10 years is right, but at least say, let's get it at least on the table and see what people think.
Dennis, to follow up on that, there was one comment in the chat by Michelle Gator, I think, about using eDNA, and the Smithsonian's advancing a big issue on that. We're hoping the IRL will be one of our like test sites, pilot sites, whatever. But following up on Linda's comments on the frequency, it was really a good point with things like oyster reefs and other things that might not be expected to change quite as frequently, like maybe once every 5 years. Like, for instance, with all the drone availability now, you could do, you know, oyster reefs and mangroves and salt marsh habitats, maybe at the northern end, and just get a lot of that habitat monitoring that you wouldn't expect to change really fast unless there was an episodic event like a major hurricane or something. And just do them, you know, 3 years, 5 years, something like that interval. And that could be coordinated, perhaps.
Well, I think these are all things that we need to think about. And I see that a bunch of stuff has shown up in the chat. A lot of people making some nice, good comments about different aspects, working with the public and emerging concerns, bioaccumulation, and so on. So I think that, you know, if you have any other thoughts, by all means, if you don't want to say them out loud, either put them in the chat or email me or David Uberger, and you know, we will add it into the mix as much as we can. And I think, as I said earlier, you know, we'll do some follow-ups with you all. We may ask some of you for some of your expertise as we try to flesh out different parts of the report. We will certainly ask all of you to take a look at whatever we think is the first decent draft that the steering committee thinks might be good enough for other people to take the time to look at. And we would hope that, you know, at least some of you would have the time and want to take a look at it and give us your thoughts at that before it gets like posted more broadly. I think you know, we really would value that opportunity. Let's see, does anybody else, Chuck or Derek or Jeff, any of the moderators, you want to say anything further about either your topics or anything else you might want to advance to the group for some thought?
This is Jeff. Dennis,
Yep.
if you'll recall, we struggled over how to make the survey, you know, completable in a timely manner. And one of those, we had a rather detailed question about habitats and how we could monitor them. So things like abundance versus diversity versus other things. And so just know that we'll be sending out something in the future that's more detailed specifically on that. And we really appreciate you filling out that survey and getting it back to us as well.
Yeah, I think that's a really good point. I'm glad you brought it up, Jeff. Yeah, we had a couple other things we wanted to do, but just logistically, you know, trying to keep everybody's time commitment down to a certain amount. And also there were some limitations with whichever survey program we went with. It worked well for some things, but not as well for others. So there may be some other things that we may just want to send out to you all and say, if you have the time to do the survey this week, please do. We're just curious to see what you think. Help us. David Huberger, you have anything to say, add?
Mostly to thank everybody for their time. I didn't realize how much information we collected until I sat here for 4 hours and watched it all be fed back to me. But thanks again, everybody, and especially our speakers today, taking the information I had and making those presentations was no small feat. So thank you again.
All righty, so last call. Anybody want to either say anything or text anything in the chat room and we'll cover it? Okay, otherwise, I hope you all have a really good upcoming Thanksgiving. I know for all of us it'll be different than normal, but it's 2020. That's the way it is. And I hope you all stay really safe and healthy and your families the same. So I think we're going to let you all disappear. We are not shutting down yet because we have to make sure that we secure like the chat room and get all that information and so on and so forth. So I just want to thank everybody for your time, and you will definitely hear from us again. We know where you are. Okay, thank you.